Good morning everyone. This is the continuation of Morphology of Bacteria. This is the second part. We were talking about the different parts of bacteria cell. We started with the anatomy of bacteria cell. We talked about cell wall, cytoplasmic membrane, cytoplasmic inclusion, and two of the appendages. One is bacteria flagella. So we'll continue with that. Next, bacterial appendages present in few of the bacteria, not in all the bacteria. That is Bacterial Fimbriae, otherwise known as pili. It's a fine hair-like structure or the surface appendage which is short, thin and straight. It is composed of a protein called pilin. There are few bacteria which possess this Bacterial Fimbriae, example Escherichia coli and Klebsiella. The function. Bacterial Fimbriae is an extra additional virulence factor which are of different types. There are actually 8 types of Bacterial Fimbriae. One is the common type which is numerous in number and it helps in attachment. There is another type which helps in conjugation which are longer called as sex pili. Now other function for this Fimbriae that act as an antigen. It induces immune response for the production of antibody, helps in diagnosis. Again it helps in hemagglutination test. Bacteria which are fimbriated shows the presence of pellicle. How do you detect Fimbriae? Bacterial Fimbriae can be detected by using electron microscope or some tests like hemagglutination test. Pellicle formation is an indication of a fimbriated bacteria. Coming to the next structure, that is bacterial capsule. Bacterial capsule is an amorphous viscid bacterial secretion. It is usually polysaccharide except in Bacillus anthracis. Bacillus anthracis is a bacteria which causes zoonotic infection, anthrax. In this bacteria, the capsule is made of polypeptide, not polysaccharide. If the outer layer is the outermost layer, it is loose and undemarcated, you can call it as slime layer. If it is sharp and defined structure, you call it as capsule. Functions. What is the function of this capsule? It mainly acts against or inhibit phagocytosis. Phagocytosis is the immune system, is a mechanism from immune system to prevent the infection. So capsulated bacteria can inhibit phagocytosis. It acts as a protective against antibacterial substance also. It helps in identification and typing of the bacteria. There are few vaccines which are prepared, that is capsular vaccines, pneumococcus vaccine, meningococcus vaccine are the examples. How do you demonstrate the presence of a capsule in a bacteria? It can be demonstrated by doing negative staining. Negative staining like India ink stain is used, where the background and the bacteria takes up the stain and only the halo around the bacteria shows that's the presence of capsule, because capsule cannot take up the stain. So it looks like a halo around the bacteria. So the whole slide and the bacteria appears dark, whereas capsule is halo. That is negative staining. Other methods include special staining, that is McFadden reaction is done in case of Bacillus anthracis for the detection of capsular material. Then we have quelling test, that is a capsule swelling test which can be done. Other serology tests also can be done for the demonstration of the capsule. Example. Capsulated bacteria example Streptococcus pneumoniae, which is known as pneumococci also, Klebsiella species, Bacillus anthracis. We have a fungus which is also an example for capsulated. So capsulated fungi example Cryptococcus. Then there is a bacteria Streptococcus salivarius which shows both capsule and slime layer. Bacteria spore. Now coming to the bacteria spore, is a highly resistant dormant state seen in some of the bacteria. So we have few group of bacteria which can sporulate during unfavorable condition. These spores helps them to survive in the unfavorable condition, and once the conditions are favorable for their multiplication, they come back to the vegetative state. Now the spore can be parts of the spore. It can be core, cortex, coat and exosporium. Based on the number of the spores, like where is it positioned or what is the shape of the spore, there are different types. For example, the position of the spore, it can be central, it can be in the center of the bacilli organism, or it can be subterminal, or it can be placed as terminal. Sometimes it is, you can see the spore is spherical in shape, or sometimes it can be oval in shape. In some bacteria, it acts as a bulging spore, whereas in some other bacteria, it acts as a non-bulging spore. Bulging spores, the diameter of the spore will be greater than the diameter of the organism. Non-bulging diameter is as equal or the lesser than the bacteria size diameter. Now, if the spore is present inside the bacterial cell, parent cell, it is known as endospore. Usually 5 to 20% is made up of dipocholinic acid in case of spore, and it contains high calcium. Example for sporulating bacteria in gram-positive aerobic bacteria is Bacillus anthracis, which cause anthrax. Then we have Clostridium tetani, which is an anaerobic bacteria which shows the property of sporulation. Gram-negative bacilli example is Coxiella burnetii. Sporulation. Sporulation takes place with 7 steps. From bacteria, when it goes to the spore stage, it happens through 7 steps. The first step is axial filament formation. There is a filament formed, that is the starting stage of the sporulation. Second step is the septum formation. After axial filament formation, it forms the septum. Third one, engulfment of the fospore. After that, it makes the cortex, that is cortex synthesis. Then there is formation of the coat and the exosporium. Later the spore mature and forms a proper spore which is released later. So the steps in sporulation, how the bacteria convert from vegetative state to spore state, consist of axial filament formation, septum formation, engulfment of fospore, cortex synthesis, formation of coat or exosporium, maturation and spore releasing. Germination. Now when the spore comes in contact with the condition which are favorable for the growth of the bacteria, it comes back to the vegetative state. That is known as germination. Spore wall is shed, spore coat is ruptured and germ cell elongate to form the vegetative cell. Demonstration of the bacteria spore. It can be demonstrated if you do a Z-N staining, that is seal-Nielsen staining, which should be modified. Modified Z-N staining, that is using 0.25% of sulfuric acid. What is the use or the application of the bacteria spore? It is used as a sterilization control. As it is a highly resistant stage, it can be used as a control for the sterilization procedures. It is misused by bioterrorism. It is misused for bioterrorism. Spores can survive in unfavorable conditions for a longer time and can be used for spreading the infection in outbreak of the infection. Coming to the other states or other morphology which are seen in bacteria, one form is pleomorphic stage. Pleomorphism is the variation in the shape and the size which are shown by some of the bacteria, example Haemophilus species. Then we have L forms. L stand for Lister form. They are the cell wall deficient forms discovered by E. Klenberger. There are some unstable forms also, protoplast, which were actually gram-positive bacteria, and spheroplast, which are actually gram-negative bacteria. When the cell wall is lost, it makes an unstable forms known as protoplast and spheroplast respectively. Bacteria growth. Now the bacteria divides by binary fission. The time taken by the bacteria to make it to 2 daughter cells. One bacteria grows in size in optimal condition and then makes 2 daughter cells. That procedure is known as generation time. It takes 20 minutes for most of the organism like Escherichia coli, but it can be 20 hours in case of Mycobacterium tuberculosis and it can be 20 days in case of Mycobacterium leprae.